Now, actually here in the textbook, right now the topic is incomplete dominance, and then codominance, and then dihybrid experiment. Actually, चलो फाइन આપણે એવી જ રીતે કરીએ. Continue with the textbook. Incomplete dominance. Actually, this incomplete dominance explains us that no one is dominant, no one is recessive. Fine. Actually, before continue this, red and white flower. Red and white flower. If you cross with the red and white flower, pink flower will be obtained. Fine. So suppose example orange color rose flower. If I want to produce the orange color rose flower, so in between which flowers you cross? Yes, red and yellow. So here red and yellow genes for that. The chromosomes are mixed together and mixture effect is created. Fine. One thing I want to tell you that in the monohybrid experiment, if you have learned that experiment properly from the textbook, in textbook it is given that blending does not occur. At the time of the monohybrid experiment, if you think about that, I have told you that tall plant cross with bud plant. So in F1 generation, all the plants are tall, and you have accepted that, sir, yes, you are right, because capital T is dominant. But one question is moving in our mind: is it possible that medium height will be opening there? Yes. So in each and every experiment, we can't say that it is definitely dominant or it is definitely recessive. Fine. So here, incomplete dominance explains us that no one is dominant and no one is recessive there. Fine, that type of the experiment can be performed here. So, according to the chart, yes, we continue with the units were there. It proved that no any character, no any trait is dominant or recessive. Why? See. Here, the incomplete dominance can be performed on the two plants. This experiment can be performed on two plants. Let me tell you the names of it. Fine. First is Evitinum species, also known as snapdragon, also known as dog flower. Fine. Erythrinium species. It is also known as snapdragon, also known as dogflower. And another Mirabilis jalapa, also known as bulbas. Fine. So these two are very important points. Fine. Erythrinium species in that, and Mirabilis jalapa, also known as bulbas. So in all these plants, these all are plants. In all plants, these two plants, this experiment was performed. Fine. Which type of the experiment can perform that? Fine. Let me continue with that. So here the flower color. So suppose example, color of flower. So here are two types of colors. Red flower. For that we give the alphabet, or we assume the alphabet, that is capital R. And for white flower, that is small r. But here remember that if you write the incomplete dominance, yes, now one question is moving in your mind. So this question will ask in how many marks? 3 marks. Fine, with the chart. Or majority. Here this question will be asked in the MCQ only. Select the proper option for Mirabilis jalapa or Erythrinium species, or snapdragon or the dogflower. So here you know that each and every word, that here we select the color of the flower in the snapdragon or the bulbous plant. Red flower is given by sign capital R, but don't write here dominant, please, because here we have to prove that no one is dominant, no one is recessive, because here blending occurs, means both the genes, the two genes responsible for different color. Here this is the one character, but these two are contrasting traits. Fine. So here for red color, the gene is now mixed with the gene which is responsible for white color. Fine. So blending occurs. So now we can select the two plants in P generation, capital R cross with small r. So here this is the P generation. Select the one plant as a female and this one plant as a male, cross with each other. So here you know that genotype is capital R and small r. So phenotype is here red color and white color. So now as you know that we have to draw the unit square there. Fine. So this is F1 generation. Unit square. So what you write here? So red flower. So cross with each other. So red flower, two gametes. Fine. So red flower, for that two gametes are there. So these both the gametes are capital R and capital R here. and here the two gametes are there. These are small r, small r. You know it very well. So here in these squares express that the type of the zygote. So you know which type of the zygote here forms. Find capital R as well as small r. In all these squares, fine, as you know it very well. Capital R, small r. Fine. So here, according to the monohybrid experiment, you think about that. This all the flowers are red in color. Fine. According to the monohybrid experiment, you think about that. This all the flowers are red in color, but it is wrong. Here all the flowers are pink in color. Why? Because you know that blending occurs. Blending means nothing but the mixture occurs. Fine. So here the gene which is responsible for red color and the gene which is responsible for the white color, both are blend together, means mixed together and form the mixture character there. So here all the flowers are pink instead of red. So here this pink color in the F1 generation, this indicates that no one is dominant and no one is recessive trait. Fine. So now as you know that what we will do, if self-fertilization occurs in F1 generation, then as you know that in F1 generation self-fertilization occurs. Then in F1 generation which zygotes are there? Capital R, small r. So capital R, small r cross with capital R, small r. So here you take the one plant as a female, one plant as a male, cross with each other, fine, and you know that phenotype is pink color. Now we have to draw the Punnett square of the F2 generation. So here as you know that this is F2 generation Punnett square. Here what we have to do? So here capital R, small r. This flower produces the two gametes. So here one is capital R and another is small r. Fine. Similarly here, this pink flower produces the two gametes. One is capital R and another is small r. Now dear all students, please understand this perfectly. Fine. So here in F2 generation this square, it is capital R, zygote. Fine. Here capital R, small r. Here also capital R, small r, and here small r, small r. That you know it very well. but understand the phenotype perfectly. So this capital R indicates red color. Fine. This capital R small r consists pink color. This capital R small r also consists pink color, and this small r consists white color. So now what you have to do in F2 generation, genotypic ratio is what 1:2:1. How? Capital R is to capital R, small r is to small r, right? So you know that three types of the genotypes are there. Capital R, that is 1. Capital R, small r, these are 2. And small r, that is 1. But phenotypic ratio, yes, you are right. That phenotypic ratio is also 1:2:1. So here what we can write? Red is to pink is to white. So here in F2 generation, out of the 4 plants, 1 plant is red, 2 plants are pink, and 1 is white. So what is the difference between monohybrid experiment and incomplete dominance? So here in F2 generation, in the monohybrid experiment, you know that genotypic ratio is 1:2:1, but phenotypic ratio was 3:1 there, and blending cannot occur. And if you have read the monohybrid experiment properly from the textbook, so in F1 generation only tall plants are there, no chance of the dwarfness, because recessive gene is there, dominant genes are expressive characters. But in F2 generation you obtain all the characters, means both the characters. Some plants are tall, some plants are dwarf also. Fine. But here at that time we say that for tall plant capital T gene, and for dwarf plant that is small t gene. Fine. Both are not mixed together. Both are segregated from each other. 2 genes are there in the plant, but both are not blending together. They mix nathi thata. Fine. But here at the time of the zygote formation, fine. So here in the F1 generation, for red color and white color genes are blending together, and as a result pink color is obtained there. So you have to draw this Punnett square of P generation, F2 generation, F1 generation, and you have to write the 3-mark question in this way, and you have to explain that.